This meeting is being recorded.
Okay, it is my delight to introduce Boris Adelessi, who's a PhD student in Dr. Yu's lab, majoring in plant breeding and minoring in statistics. He holds a BS in agronomic sciences from a university I am not going to try and pronounce in Benin, and a master's in plant breeding and seed systems from another university I'm not going to pronounce in Uganda. And I was fascinated to learn that he worked at the Regional Center of Excellence for Crop Improvement in Uganda, where he led a project on selecting resistant cowpea varieties that would be important for Benin. So today he's going to talk to us about his research with Dr. Yu, which uses UAVs to look and understand the genetics underlying sorghum plant height. So Boris, the floor and screen are yours.
Thank you, Jamie. Can you hear me online? All right, nice. So yeah, welcome everyone, especially those of you who could make it in person. We have quite a number of students here, so that's really good. You're not just alone. And thank you also for joining online to attend my talk. That will be on using UAV imagery to investigate the dynamics of sorghum plant height genetics. So let's get started. All right. Yeah, so we've made some good progress recently in genomics, and that has greatly improved our understanding of major agronomic traits, genetic basis. But at the same time, the low throughput of manual phenotyping has remained a bottleneck. And an example of this is plant height, where typically we measure the final height, but we actually know that this trait is dynamic. And it is controlled by multiple factors that are occurring throughout the whole developmental process. So genetic studies based on only the final height just actually show us a snapshot of the whole complex and dynamic process that led to that final height that we see. So it is therefore important for us to uncover the genetic basis underlying these temporal dynamics that we see in plant height, because not only it will help us to enrich our understanding, but also to be able to better connect genotype to phenotype. On this figure here, we can see a recent study on wheat that also focused on the developmental process, and they identified 3 types of QTL. The first one is a transient QTL. And a transient QTL is a QTL that is detected only during the early phase of the growing season, but then disappear later on. The second type of QTL is a stage-specific. And a stage-specific QTL is the one that is detected at a particular developmental stage during the growing season. And finally, we have what they call a persistent QTL, which is detected more or less throughout the entire growing season. So having now made some good progress with high-throughput phenotyping, I think we have the opportunity to be able to now collect a time series plant height data at a large scale with high efficiency resolution, but also at a low cost. So this work have multiple objectives, and 2 of them that I'm going to present today is first, we try to use the UAV-based RGB imagery to estimate sorghum plant height on the time series, and also use this time series data to be able to investigate the dynamics of sorghum plant height QTLs across developmental stages. And to do that, we use 2 genetic populations. The first one is the recombinant inbred line population, TX430 by P898012. And this population has actually been used by our lab, and you can see on the lower bottom picture here, it has been previously used in our lab to dissect 2 plant height QTLs, namely DW3 and QC7.1 on chromosome 7. And the second population, which is on the picture on the right, is the sorghum association panel, which compared to the reals have a broader genetic base. So we planted this population out in the field, the reals for 3 seasons, and the SAP in 2019 and 2021. For each growing season, we conducted overflights at multiple developmental stages, as we can see on the lower bottom on the left bottom figure here, where we cover the entire growing season at different growth stages. And we took aerial images of the field using a Canon camera that was mounted to a DJI S900 drone. And to be able to rectify the pictures later on, we lay out 9 to 12 ground control points, depending on the size of the field, and that we georeference using an RTK GPS. So from each image collection, we had to process and be able to extract features. First, we use a structure from motion software called Agisoft MetaShare, where the first step consists of aligning this picture and be able to discard those with lower quality. And after that, we now import the coordinates of the ground control point, and that helps us to rectify, as I mentioned before, these images. And after that, we are able to build a dense point cloud that, if you can see here at the bottom, and we have on top the waypoints in the field. The dense point cloud enables us now to build the auto mosaic as well as the digital surface model. So the digital surface model has both the elevation of the ground as well as that of the plants on top. So what we did is that using the first overflight, which had the least vegetation cover, we were able to interpolate the background elevation to obtain a digital terrain model, which is now the elevation of the background. And we can now subtract the DTM from the DSM to obtain what we call the crop surface model, which ultimately helps us to estimate plant height on plot basis after segmentation. So for the real population, we are now interested in knowing how does this UAV estimated height compare with manual measurement that we had taken at each overflight. So as we can see on the two scatter plots on the left, both in 2018 and 2019, there was strong correlation between UAV estimate and manual measurements. And from the broad sense, from the variance component analysis, the broad sense predictability on entry mean basis showed that up to 87% of the phenotypic variation observed were due to genetic effects. So from this result, we thought, okay, the 3D reconstruction of the field was able to successfully capture the genotypic differences in height. So from this on, for the repopulation, subsequent analysis focused mainly on the UAV estimated height. First, we use a logistic function to model the growth trajectories of each of these genotypes. And you can see that on the left. And taking the first derivative of that function, we were able to obtain the growth rate or rate of change throughout the growing season on the right. And from the growth curve, we can see that, yes, as expected, the phenotypic variance increases as the season goes. And we also saw some transversal segregation from the rails, which met our expectation from what we saw in the field. So now the next step is to answer the question, are there some QTLs that influence the whole growth curve as a whole together? And having done a composite interval mapping, we found that QTL colocalizing with DW1, 3, and QHT 7.1 actually affects the whole growth curve. But now this one does not tell us yet what happened at specific growth stages during the season. So from the logistic function, we were able now to estimate a plant height at 5-day interval for each genotype, from 20 days after planting all the way to 100 days after planting. And if you are able to see this figure, it might look a bit small, but panel B here. So we use this model estimate for plant height and then conducted again QTL mapping throughout the season. And from what we saw, early during the season, we observed up to 3 QTL that were transient on chromosome 4, 5, and 7. And these QTL were detected at early season, but then disappeared later on. But a bit later on, we saw that QTL that are co-localizing with the major gene that we saw before that we've seen before, DW1, 3, and 367.1, were now able to be detected from mid-season and were persistent till the end of the season. We did a similar analysis for the growth rates. And similar to what we saw up, we also detected two QTL on chromosome 5 and 6 that were transient. But later on, the major QTL showed up, and mainly DW1 that extended longer. So at this point, we are now going to switch to the sorghum association panel. For the sorghum association panel, we were not able to take manual measurement at every overflight. So we only compare the final measurement for both UAV and manual measurement. And again, the correlation was strong. So that was good news. And using the similar procedure, we tried to model the growth trajectory. But what happened in 2019, due to some reason, the first overflight was at 42 days after planting. And some of the genotype having a broader genetic base, some of them had already reached the final heights by that point. And we can see those ones here that are very short. So we didn't capture very well. And the good news is that in 2021, that taught us a lesson, but now we're able to increase the temporal resolution. So last summer, we were able to start flying very early during the season to better capture the growth trajectory of those short genotypes. But we still went ahead and already run some GWAS analysis of the UAV estimated height. And as we can see here, we detected, again, known plant height genes, loci, DW1, 2, and 3. And compared to DW2 and 3 that were detected, so that were persistent throughout the whole season, DW1, on the other hand, was not detected during the early phase of the season, but only later on during the season. And also, we found a stage-specific QTL, potential QTL on chromosome 2 that were only detected later on during the later developmental stage. So, in conclusion, from the UAV RGB imagery, we were able to successfully estimate the plant height with relatively high accuracy. And having done the QTL mapping of this UAV estimated height, we were able to detect a strong genetic signal with changing effects. So we think that the high spatial and temporal resolution of the UAV imagery can be leveraged to further decipher the genetics of sorghum plant heights. So as I mentioned before, this is just a portion of one of my projects. So we still have some ongoing work. So we are currently processing the images from the overflight in 2021. And we are also investigating these same dynamics in maize. So we are working on 2 crops at the same time. And yeah, to make it more enticing, we are also now designing some experiments where we can combine a gene expression analysis to be able to profile the expression of the genes that are within these QTL regions throughout developmental stages, also to see what is actually happening at the expression level. With that, I would like to thank my major professor, Dr. Jamin Yu, for his great support since I've been here at ISU, and all our lab members for very engaging discussion that we have during lab meetings. Thank you for providing me with a very friendly environment. I really appreciate that. And I would like to also acknowledge previous lab members that have paved the way for us, especially Shin Lee, as I mentioned, who already used this population before us, and my committee members. And I would also like to thank the undergraduate students who have been very dedicated and have helped us during each summer to collect data. It can be quite intense. And I would like to also thank our collaborators. And this study was possible thanks to the funding from the Iowa Crop Improvement Association, USDA NIFA, the Department of Agronomy, and the Plant Science Institute. With that, thank you for listening, and I welcome any question. Thank you.
Okay, we'll play the same rules as before. If you have any questions, raise your hand. If you're in the room, shout it out. We don't know, so somebody tell us that there's a question coming from the room. I guess I'll kick us off. Boris, when you're these two new QTLs that you've identified on chromosomes 5 and 7, maybe. I don't know much about sorghum genomics. Is there is the genome sequence good enough that you can look and see like what, if any, candidate genes are underlying those? And if so, have you?
That's an excellent question, Jimmy. And yes, we do have the sorghum genome being sequenced, and we're able to look into that. And that's what we are doing. And that's why we are hoping to now incorporate gene expression analysis. Nice. For those particular region, yeah.
Nice. Michelle?
Okay, very nice talk. I have 2 questions. My first question is, earlier on in your talk, you had a slide where you compared the 2019 season to the 2020 season, and you had the 3 different correlations. And what I noticed was that in the 2019, the correlations were all kind of tightly clustered, overlapping. But in the 2020 year, the correlation, which was the earliest one, seemed to be physically separated from the other 2. And I wondered why was that? So yeah, the slide exactly. Why is the green, kind of overlapping the red and purple on the top, but not on the bottom?
That's a very good question. So for 2018 and 2019, if we look at 2018, the green dots were plant height that were estimated at 42 days after planting. So that's a good point. While that of 2018 is at 50 days after planting. So there'll be more variation that was observed. Yeah, that's why we have the 2018 being very clustered at the bottom, because there wasn't much variation at that point compared to that of 2018. Yeah.
And so then my other question was, in your Manhattan plots for your GWAS, sometimes you see QTLs where you have a lot of like, you have a peak of dots. And sometimes you see QTL where it's just 1 dot. And what's the difference, and does that impact how you interpret the impact of the QTL?
That's a great question. So yes, there are some loci where we see multiple dots like here, compared to like on chromosome 2 when there is only one dot. Yeah, so my attempt to answer that question would be that within that linkage block, there are multiple SNPs that were able to pass the threshold, compared to this one where there is only a few SNPs that were able to pass the threshold.
And so does that bias how you interpret it? Like, do you consider them the same? So like DW3 and DW2, at least at 106 days, look very different. Do you believe them both equally?
Pardon, come again, please.
So does that impact how you interpret the strength of the QTL or the confidence in the QTL?
Oh, okay. Yeah, so the question is that does that affect how we interpret the strength of the QTL, right?
Mm-hmm.
Oh, okay. I see. Possibly yes. Yeah, it may definitely affect how we interpret the strength. Yeah, because multiple SNPs showing up indicates a stronger signal. Yeah. So that's why we have to look at like this one snip here. We'll go and look at the genome, what is in that region. Yeah, so we want to confirm. Yeah, that's a good point.
Thank you.
Any other questions? Any questions from the room?
No question.
Okay. Well, then let's give our speakers just one more round of applause. Thank you guys so much. These were fantastic. Thank you for taking the effort to set up the presentations in the room so you could have your full presentation experience in front of an audience. And we will see you guys. No. Do we have seminar next week, Xiaoming?
You're muted.
Yes, we'll have a Zoom seminar next week. Yeah.
Okay. All right. We will see you next week then.